Good morning and welcome, everyone. It is hard to believe, but this is our last webinar of 2025. Don't worry, we've got plenty planned for 2026, and we're going to end the year strong with not one, but two amazing researchers today. And they're going to be talking about a very important topic in the field of produced water treatment and reuse, which is whether or not there are any negative or undesirable impacts when irrigating crops with treated and purified produced water. So joining us today are Adrianne Lopez, who's the Research and Development Manager at Texas Pacific Water Resources, and Punhasa Senanayake, a postdoc researcher at New Mexico State University in the Civil Engineering Department. Together, they bring a wealth of experience in produced water treatment, and I know for a fact that neither of them are afraid to get their hands dirty piloting these next generation water treatment systems. Today's webinar is going to be building off an awesome and recently published paper by this fantastic duo that's titled Plant Growth, Ion Dynamics, and Microbial Communities in Soil Irrigated with Treated Produced Water, I'm sorry. This paper is open access and the link will be in the description below. But without further ado, Adrianne and Punhasa, please take it away. The virtual stage is yours.
Thank you, Zach. Hello, everyone. Good morning, and thank you for joining today's presentation. I'm Punhasa Senanayake from New Mexico State University. I'm here with Adrianne Lopez from Texas Pacific Water Resources. Today, we'll be sharing our findings of our greenhouse study, which was irrigated using treated produced water at different salinities, and what does that means to the soil health, ion transport, microbial community, and plant growth. So this work was associated with New Mexico Produced Water Research Consortium to provide objective scientific-based information rather than promoting a single technology. So water scarcity is one of the bottleneck for all southern states, especially arid and semi-arid region, namely Arizona, New Mexico, and Texas, which runs on tight water budget. So all these states use brackish groundwater, vary between 40 and 55% for potable and non-potable use. In irrigation, this water is part of the cycle. As you can see, it helps back to recharge the aquifers. But however, the deviation that undergoes through desalination for potable and non-potable water use does not directly helps to recharge the aquifers, which we need to rely on the precipitation. However, with the population growth and economical development, the branch that deviates from this cycle has skyrocketed. Therefore, there's huge demand for water recharge, because if it's not properly recharged, the water comes out from the aquifers are highly saline. Therefore, it influences the crops as well as the technology that implemented to desalinate these waters. Therefore, communities have recycled some of the waste streams, namely municipal wastewater, brine waste, and seawater, to fulfill this gap between the demand and supply. And these waters can not only be utilized in agricultural application, but also industrial application, such as hydrogen production and chip manufacturing, where even though there are strict water quality requirements, the regulations are slightly loose. In addition to that, there are a couple of plants that already utilize these waste streams to fulfill the water demand for potable and non-potable application located in Florida and California in US. However, the newest member of this family is the produced water, which is the waste stream from oil and gas that is currently recycled for the in-house application, but has a potential to reuse in these non-potable application. However, the compounds and its composition and the concentrations of these produced water becomes challenging to recycle this water for this application because the water quality strictly depends on the geological formation, chemical additives during the hydraulic fracturing, and the treatment train itself. So cumulatively, these factors challenge the recycling produced water for fit for purposes application. However, there are a couple of studies that was deployed using raw produced water, dilute produced water, and treated produced water for agricultural application. Among these findings of these studies, we can see that higher TDS treated produced water increase the sodium, chlorine, and sulfate concentrations, which elevate the sodium absorption ratio, which is a key factor for irrigation, and also disturb the fungi community. In contrast, when the TDS of the treated produced water below 1000 milligrams per liter, it has shown that it maintains the organic carbon and balance calcium magnesium ratio, which helps to maintain the soil structure and the nutrition available in these soils. However, these studies are scattered throughout different papers, as you can see in the bottom. Therefore, there are next to no studies that evaluate all these parameters all across the board for treated produced water. However, all these studies illuminate the fact that raw produced water or dilute produced water is not an option for agricultural application. Therefore, we partner up with Texas Pacific Water Resource for deploy a study to investigate all these factors, which includes ion transport, soil composition, microbial community study, and how the plant growth affected by these treated produced water with different salinities. So to discuss more, to discuss further about the project goals, the treatment train, and the experiment setup, Adrianne will continue from next slide onwards. Adrianne.
Thank you, Punhasa. So I'm going to talk a little bit today about the treatment train, how it works and how it was developed, kind of why we added different steps where. We started working on produced water desalination in 2022 and brought this pilot to Midland in 2023. The pilot that you're seeing pictured in the right-hand side of that slide is Texas Pacific's freeze desalination unit. So Texas Pacific utilizes freeze desalination in conjunction with reverse osmosis, requiring some pretreatment. So we've treated for reuse with an oil and gas now since 2017, are very familiar with the flock and drop process, utilizing oxidants to assist in oil separation, oxidation of iron, removal of H2S, and then coagulation processes. So in this trial, we used an aluminum oxide powder and a membrane combined with a membrane process because we really had a desire to have turbidity below 1. So traditionally, when we don't use a membrane associated with that pretreatment process and we just have oxidation, coagulation, flocculation, and removal, sometimes you do still have some fine colloidal particles floating around. Those are important to remove from before freeze desalination because they create additional nucleation sites for ice to grow. So that's something that we want to be conscious of going into freeze desalination, is having oil below 5 parts per million and having turbidity below 1. Going into freeze desalination, the unit that you're seeing here operates by recovering about 50% of the water in the form of ice. So you can see the very far right picture that looks like a tiny little O, that's actually the ice forming on the inside of those tubes. So the fundamentals that we use here are that brine has a lot lower freezing point than water. As we build that ice crystal, if we build that crystal slowly and in sheets, it actually does push the salt out of that crystal structure. So what you're left with is ice that has about 50% less solids that can then be melted and on to the next step. So freeze desalination, the recovery, the volume recovery is based on the starting salinity. We can only concentrate up to about 220,000 parts per million in the brine. So we're able to recover from the starting produced water is dependent on that. When we are now under construction for a larger facility in Orla that is also using freeze desalination, but we have also done a lot of research and had some advancements in different types of ice. So this unit that we used during our very first pilot, we were only able to make this one form of ice on the walls. We're now able to make a couple different kinds that make recovery and harvesting of that ice from the fluid quicker and less of a batch process. So freeze desalination is very effective until you start getting down into the lower 10 to 15,000 parts per million waters, which is why we then follow with RO. RO is also very effective. It's tried and true to remove a lot of what we see in produced water. And so we employed RO as our final step along with activated carbon post RO. We had activated carbon pre-RO as well as green sand. And then during wet tests, we're still having some issues on the Ceradaphnia. So we decided to add a activated carbon post RO, and that seemed to help a lot. We didn't have any issues after that. Next slide, please. The study was done with soil taken from counties in the Permian. So we took soil from Texas Pacific lands in Reeves County and Loving County, anticipating that those are the two counties that were going to be, in need of an alternative solution the quickest. So we went out to those areas, tried to find relatively undisturbed areas that we could collect soil from, did some surveying of the native plants, and then decided ultimately on the native plants mentioned here. So you see, Mexican feathergrass, yucca, mesquite, and then we also used alfalfa. We chose alfalfa because it uptakes a lot of water and it is very tolerant to West Texas climate and climate variability as well. Next slide, please. The way that we laid out the study in the greenhouse, so there were 12 different greenhouse boxes. The top row had what CB stands for cowboy. That's going to be our Reeves County soil. The next row is the whiskers. That's going to be our Loving County soil. Those top two boxes, or those top two rows had native plants and were irrigated with different salinities based on the vertical row. So horizontally separated by soil type and plant type, vertically separated by water quality type. So the water qualities that we chose were 15,000, 1,500, and a control, which was just, it was Midlands groundwater that had been processed via an RO to supply to a nearby man. So that water had very, very low solids. It was about 15 parts per million of TDS. And we irrigated these boxes for about 6 months to assess, differences in growth across the boxes, differences in soil health. On the alfalfa, we were taking plant samples to do forage analysis. And each box was 2.5 feet wide by 7 feet long and 3 feet deep. So this gave us quite a bit of soil to analyze. We could go all the way down to the 3-foot depth of the soil column and assess, the effects that one, the amount of water had, and then two, the type of plant had. So alfalfa has very different root systems than native plants, and it also takes a lot more water. So we were able to not only assess, the application of water, but also the application to different soil types, different water types, and different plant types, all of which we could share with New Mexico. Next slide, Punhasa. These are some photos of the greenhouse while it was active. So something to point out, the alfalfa has a lot more sprinkler heads in it than the native plants. So the native plants received about 15 to 20 gallons of water a week, and the alfalfa received about 50 gallons of water a week. So all of this took place in the same spot. This layout here just kind of shows what our R&D facility looked like during piloting. So we had a laboratory on site as well. equipped with ICP and IC. That allowed us to get instantaneous results for our water quality testing. We also had the capabilities of doing a lot of different, DO, pH, TDS, things like that, and then some soil tests too. So all of our data was validated via third-party lab. We had third-party sample collection for our irrigation tanks as well as our soil and our plants, and then we were also internally taking regular plant and water samples just kind of to assess the crop over time. Next. And I'm going to discuss just some high-level results of what we saw, and then I'll let Punhasa get into the bulk of the data, which was very exciting. So because alfalfa uptakes so much more water, we decided to focus specifically on alfalfa for this study. So alfalfa was, one second. So alfalfa was chosen because of the amount of water that it uptakes, and because we could grow it in West Texas. It's also a valuable crop. So all of the analysis that we're going to talk about in the next couple slides are going to be referencing specifically those alfalfa boxes. While we did have a lot of analysis on the native plant side, we just decided to use those because we felt like the data was more robust. But going forward, Texas Pacific Water Resources will be also participating in a large rehabilitation project associated with our next desalination pilot. So just a little bit more detail about those soils. So Reeves County soil was a lot more clay rich, didn't have as good of drainage. The Loving County soil was very sandy, had very good drainage. We saw a big difference in the plant health based on those soil types. So what you can see in the data to the left, so what we're looking at, RFV stands for relative feed value. The boxes are separated by the irrigation quality and then the county. So what you can see is Reeves County had very good quality alfalfa in the higher salinity boxes. And then Loving actually grew the best alfalfa at that 108 or that 1000 TDS range. What we noticed too in doing this project, we did not use any fertilizer or pesticides or any soil additions here, with the exception of some bacillus-based armyworm deterrent. We did have plenty of pests present during this greenhouse experiment because of the lack of the use of pesticides. But what we noticed in the Reeves County soil was that because it was a lot more rich in minerals starting out, it did actually grow better alfalfa for longer durations. So the Loving County soil did well in the beginning, but quickly that soil became leached because it just didn't have a lot of minerals in it. Whereas the Reeves County did sustain alfalfa for longer. Something else that we saw was that in the higher mineral content waters, we saw a healthier crop, and that was because we were adding back a small portion of our RO concrete to obtain that 1000 and that 1500 TDS goal. Now, what we learned in doing that is that that's not a feasible way to increase TDS. Well, it did make healthier crops because of the addition of some minerals that would have been present in fertilizer. We did also see increases in certain metals in the soil because of adding that RO concentrate back without conditioning it or extracting certain minerals from it. So all this tells us really is that, if we're growing alfalfa, we do still need to add minerals, which is why you see a healthier crop in those higher salinity boxes. But as Punhasa begins discussing the soil, as you can see in the next box, the sodium absorption ratio actually increased by 1/4 the addition of the 100 box. And the chloride increase was significant as well. Let's see. The room was kept steady, and at night it decreased to about 85, and we kept the humidity in there slightly less than 35%, with the exception of after we would have an irrigation event. So sometimes that would increase the humidity, but we did keep 2 large industrial dehumidifiers there to mimic what we were seeing in Permian conditions, which is about 50 to 60% evaporation over a 24-hour period. And with that, I'll let Punhasa dive into the results.
Thank you, Adrianne. Yeah, with that, if we first let's look into the water profile a little bit in detail that was used in these studies. So as you can see, and as Adrianne highlighted, we use a range around 1,500 milligrams per liter, 1,000 milligrams per liter, and 500 milligrams per liter of treated produced water, and a control system as well with around 12 to 15 milligrams per liter. So the parameters that links with the TDS also show the same trend. For example, the conductivity. The SAR number was calculated in different manner, different way. I'll show you how it was calculated, but it is simply a ratio between monovalence and divalence. As you can see, a better balance can be seen in control water, but less amount of ions are present in these waters. Even though the RO rejection was blended back with RO permeate, the organics are at the lower end of the water, and mostly the water was governed by the inorganics in the system. So we collected the soil sample before before the experiment, and after 40 weeks at 1, 2, and 3 feet for both cowboy and viscous soil type, and studied physiochemical properties, including conductivity, ammonium nitrogen, total solids, and 12 including 12 more other parameters. We investigate cations, anions, radionuclides, metal and metalloids, collectively cumulative to 86, which includes radium, boron, and chlorine, which are challenging for the irrigation, and organics, over 300 organics, which spread out over volatile, semi-volatile organics, and as well as PFAS. Yeah. First, we try to derive a parameter which can study the soil in macroscopic view. Therefore, we define the soil quality index here, which carried two components, one focusing on the chemical component of the soil, while the other carries the physical component of the soil with weighted average bias to chemical composition. To derive the chemical component of the soil quality index, we use pH and the conductivity. For physical factor of the soil quality index, we use total soil and sodium absorption ratio. As you can see, it's a combination, it's a ratio between monovalence and divalence. If I highlight again, the abbreviation CBA stands for cowboy soil planted alfalfa, and viscous A means viscous soil planted alfalfa. 1, 2, 3 is the treated produced water, while number 4 is the control system. The TDS and the number here has an inverse relationship with what this means, that the one here was irrigated using 1,500 milligrams per liter. Here it's number 2 is 1,000 milligrams per liter, and number 3 is 500 milligrams per liter TDS. So overall, when we looked into the cowboy soil, we see after 40 weeks, there's a decrease in soil quality index, and the lesser decrease can be observed for the soil that was irrigated using 1,000 milligrams per liter TDS, followed by a close second for control and the 500 milligrams per liter, and the worst case scenario was observed at higher TDS system, which was irrigated with 1,500 milligrams per liter. In contrast, the viscous show enhance performance, and it has an inverse relationship to the TDS, except for the swipe that we see here between 1000 and 1500 milligrams per liter treated produced water. If we look more closer into these changes with the depth, we can see a chemical component of the soil quality index fluctuate greatly compared to the physical component of the soil quality index, which was only vary between 0.01 and 0.03. And the top soil shows a significant variation compared to the ones at the bottom. This could be account to the fact that the irrigation was conducted using sprinklers, which is close to the surface. But as we can see, the physical component is buffering significantly. This could be due to the fact that the sodium is getting buffered by the soil, or it can undergo a different mechanism, which will be looked into more detail when we looked into the individual soil composition. So moving on to the ions, for cowboy soil, there are two types of ions which are which are not present or close to lower concentration in the pristine soil, and throughout 40 weeks it start accumulating in these soils. And the second set of families of ions which was originally in the soil, and we'll see some fluctuation through after 40 weeks. So first we can look into the ions that was at a lower concentration or not detected initially. Among them is the sodium that you can see which increase quite a lot and has a linear relationship with the TDS of the irrigated water for the topsoil. And when we go to the bottom layers, we can see this difference get narrowed, and the fluctuation happens within 35 milligrams per kilogram. This can be associated with the mechanism or like the soil type, because cowboy soil is clay-like soil, therefore it has the ability to undergo ion exchange properties, so it can retain more ions at the top compared to the bottom, because of the irrigation site being at the top. Other than that, we see boron, cobalt, nickel, and beryllium as well. Interestingly, there are some ions that was below detection limit in the feed water, but was observed in the soil after 40 weeks, namely molybdenum, nickel, and vanadium. Again, this can be associated with the fact that we blend the RO concentrate with RO permeate to adjust the TDS of the water. When we looked at the ions that was present in this pristine sample, and when we compare it with the 40 weeks, we can see calcium and magnesium, which are essential essential ions for soil and plant both, show a decrease for both top and bottom soil, and the ones at the bottom shows a significant reduction compared to the top one. Again, this can be associated with the ion exchange capability of the clay-like nature. Their topsoil absorb or intercalate the divalence to retain its structure, while towards the bottom there's lack of calcium and magnesium, leading to significant depletion of these divalent. However, even with these significant decrease, we can see that the concentrations of the calcium and magnesium are quite high, which buffers the sodium absorption ratio, which cascade into lesser fluctuation in physical component of the soil quality index. In addition to that, we see the potassium behave quite similarly, where when there's higher TDS, it has a relatively high potassium concentration. So topsoil retain this potassium, while towards the bottom there's less amount of potassium. Therefore, we see a depletion after 40 weeks. When we look into the anions, we see that in general, most of the ions increases after 40 weeks, except for the chlorine that shows relatively stable concentration after 40 weeks. Therefore, it is something that quite interesting to see in cowboy soil, that the chlorine concentration can be maintained for alfalfa if we irrigated with cowboy type soil. On the other hand, sulfate increases significantly, and initially these concentration was below detection limit or lower concentrations. That's why you don't see the percentage change, instead the final concentrations. Moving on to viscous soil, we see the same quite similar trend, that the sodium retain in the soil. But if we looked at the top and the bottom soil, we see a lesser fluctuation, where initially we saw for cowboy soil with higher TDS, the sodium concentration can go up to 400 plus range, but here it's around 260, and towards the bottom it's 120. So the variation is less, and thereby the ions we introduce evenly distribute throughout the soil. However, similar to the cowboy soil, we see foreign ions accumulates over time, even though these ions were below detection limit in the feed waters. Interestingly, the divalents show a quite different trend, where we see a depletion at the top, and these ions accumulates at the bottom. This is again linked with the soil properties, where viscous soil is sandy, therefore it can leach these ions to the bottom. So it shows us that based on the soil type, we need to tailor the water and as well as the irrigating frequencies. The remaining trends falls the same, similar as calcium, and this trend can be further validated with tailored water quality and irrigating frequencies. The difference between cowboy and viscous soil treated planted alfalfa is the chlorine. Here we see chlorine accumulating throughout the soil bed, which illuminate the fact that the viscous soil evenly distribute the ions compared to the cowboy soil. Looking at organics, even though we study a spectrum of organics, we only observe oil and grease and TPH, and the high and low TPH is below 8 and 3 respectively, and most of the organics are associated with oil and grease. And all these organics are below 130 milligrams per kilogram. The quite interesting and contributive finding is that the control system also shows oil and grease as well as TPH in their system after 40 weeks, which could be associated with cross-contamination or presence of these ions, presence of these organics in the pristine soil, because these soils were collected from the Midland area. Afterwards, Dr. Yang Yang Chang conduct a microbial community study. Here, we can see that uh, both fungal and bacteria maintain a rich and diverse trend throughout the samples, except for the viscous soil irrigated with 1,500 milligrams per liter, where we see a reduction in fungal in this soil. Additionally, we see the bacteria has more robustness to the TDS, but collectively we see that better performance when the system was irrigated with 1,000 milligrams per liter treated produced water. Finally, we looked into the plants as well. Here we see that we studied beyond these parameters, but to highlight the fact that treated produced water with 1,500 milligrams per liter shows a better performance all across the board, with the exception of digestibility, which shows a better performance with 500 milligrams per liter treated produced water. With that, if we look into the conclusions, we can see that the soil quality index we derive shows macroscopic understanding to filter the soils to say which one performs better. But in general, we see that TDS below 1,000 milligrams per liter treated produced water perform well for both cowboy and viscous soil type, which was rich in clay and sandy properties. For viscous, the highest performance was the control of the 500 milligrams per liter treated produced water. So sandy soil can be greatly benefit by irrigating using lower TDS systems, while cowboy soil can be benefit using irrigating with 1,000 milligrams per liter treated produced water. However, we see the calcium, magnesium, and iron decrease in most of these cases, which shows that remineralization is quite important, as well as potassium in most of the cases. Organics are pretty stable, which can be associated with the fact that initial feed water has lower TOC, as well as the microbial community contribute to maintain some of these organics as well. When we looked into the microbial study, we saw that both bacterial and fungi maintain their diversity and the richness, except for the viscous soil that was irrigated with 1,500 milligrams per liter. Looking at the plant qualities, we see that the treated produced water with 1,000 milligrams per liter show a better balanced performance, while for viscous soil, 500 milligrams per liter show better digestibility. However, with this study, we do notice maintaining a low TDS is quite important, and remineralization and maintaining the SAR number to a specific range is quite important in the long run. And monitoring and measuring the conductivity and the SAR number with some of these cations and anions can be greatly benefit for the system. And similar in the same sense, avoiding higher TDS treated produced water can benefit both the soil composition and the microbial community as well. However, since this is a greenhouse study, when we go to the field, it might change the results. But at the moment, we can see that the treated produced water can be an option for irrigation. However, when we optimize these waters, we need to consider the water quality, irrigating frequency, plant type, and soil, and need to be optimized these parameters collectively. With that, I would like to acknowledge all the contributors, co-authors, and New Mexico Produced Water Research Consortium for giving us this opportunity. As Zach mentioned, we published this work a few months back. You can follow this link or simply scan this code to access the paper, and it is openly available. With that, thank you all for listening. I'm happy to answer any questions. Thank you.
Awesome. Thanks so much, Adrianne and Punhasa. Really, really great presentation covering really, again, an extremely important topic. Don't see too many questions in the Q&A quite yet, but maybe I can kick us off with an opening question here. What is the main insight from the study you think would matter most to the public?
From my perspective of view, Zach, that treated produced water can be tailored better and can be introduced to these plants. So based on the plant type and the soil type, we can basically tailor the produced water, like the treated produced water, which can be potentially used for the irrigation. So we can retain some of this brackish water for more, like for potable water use. So we don't need to sacrifice much for the irrigation purposes.
Thanks,
And
Punhasa.
I'll second Punhasa. Oh, it's okay. I'll second Punhasa in that as well. That I think one of the most important things we wanted to take away from this is that irrigation with treated produced water is absolutely possible. And I think produced water does present us with a very good opportunity. But we do also have to be conscious, especially in more in an agriculture setting than in, a rehabilitation setting, of the remineralization that is required for these crops. One, to protect the soil, and then two, you don't, grow a whole lot of crops that don't hit the minerals. So that's just what.
Thank you. Joe's been waiting very patiently with his hand up. Joe, go for it.
All right. I have a few questions, but they don't have to be done all at once. First question, please. Regarding the better performance of the plants at, say, 1,000 TDS, what would you folks think is the driving force behind that? When I looked at the data just here for the first time, and I was trying to keep up with you folks, I did see ammonia being present in that treated produced water, and it did increase as we moved to 1,000 and 1,500. Is the ammonia having a primary role here, or is there something else that's showing up in the TDS that is important for the nutritional health of the plants?
Yes, thank you for the question. So yes, the ammonia does play, we think does play a role, but more than that, the calcium and magnesium of the soil do significantly influence the performance, because as you can see, even after about a 60% decrease over time, still the calcium concentrations are pretty high. So I think the divalence that originally was in the soil was buffering the performance, like help to maintaining the performance and buffering the depletion. So in addition to ammonia, the divalents are quite important as well.
Good. Yeah, appreciate that. Thank you. I'm also curious about the control, the comparison of the control. I'm not sure if you described much about that. What I was curious about is there are various qualities of irrigation water used throughout the country. One of the things I've heard is that we are really critiquing very strongly the treated produced water, versus what is used for ag waters elsewhere in the country. Could you folks comment on the quality of that control water versus, to the best of your knowledge, the quality of irrigation waters used in elsewhere in the Permian or in the U.S.?
Yes. Yeah,
Go
go
ahead,
ahead.
Punhasa. Well, Joe, I can say that in this study, yes, we did use very, very clean water in comparison to what is used standard around the Permian. So I visited a few farms in the process of learning about alfalfa. One of them was using a 3,000 TDS water. The other one was using a 5,000 TDS water. What we learned is that the farmers out here, because that's the water they have access to, they do actually have specific seeds that are tailored towards those salinities, which keeps the crop healthy, but doesn't necessarily preserve anything about the soil. So the study that we did after we finished in the greenhouse was you actually relocated it outdoors and irrigated it with Midland's groundwater. Midland's groundwater was about 650 parts per million in that area, and the results were very, they were actually the plants were very similar from the 650 TDS to our about 300 TDS treated produced water. I think if we would have been higher than that in our Midland control, if we would have been closer to 1,000, which would be made up of quite a few salts and minerals, we probably would have seen positive impact to the plants because we were not using fertilizer in that study again. But the farmers that I met with that are using that higher salinity, in the 3,000 to 7,000 range, long term, it's absolutely destroying their soils.
Excellent answers, you guys. Appreciate it. I'll ask one last question and back off so other people can jump in. Based on what you just shared, Adrianne, and thinking about what was said just a little while ago about remineralizing the water, to contain costs, would it be fair to say we really don't, the farmer would not have to really remineralize if he just took the treated produced water? Would it not be fair to say they'd be eons ahead of where they are today, and remineralizing could draw benefits, but they wouldn't have to?
So
Do you want that one? Okay, go
yeah,
ahead.
so the issue is that at the moment the soil by itself has a significant amount of calcium and magnesium. After 40 weeks, we see a decrease, but still the concentrations are pretty high. But if you use the same soil for like decade, these ions will be depleted pretty fast. Therefore, the plants won't be in better shape towards the end of the study. So remineralize it in short time periods, this can be bypassed, but for longer agricultural application, I think it's better to remineralize the water. The good thing is these ions are there in the produced water. We just need to valorize them properly, so we don't need to like externally introduce them. We can just get these divalents in the produced water to introduce to the treated produced water.
And I appreciate that. And there's two reasons I bring that up. One is cost, right? If the farmer can avoid it, and highly unfortunate. I think a lot of us in the industry know that, there is somebody that's been awarded a patent for the purpose of deriving an income on something that was rather obvious, and that is how do you adjust the quality of the produced water, treated produced water, to render it more favorable for the planting purposes. And as long as the farmer can avoid that, then he wouldn't be infringing upon this patent that many people feel is, well, I don't even know what to call it, just to say it's just inappropriate. But I didn't want us to all walk away from this that the water has to be mineralized. It would be a good idea, or maybe it could be handled by fertilizer itself, and that's why I raised the question. But I thank you for the answer. I'll stand out.
Thank Yeah, so in this study, we didn't include any fertilizers, pesticides. So that's something we need to add upon this. So I do agree. So if there are sufficient divalents and the ions that missing in the water can be introduced via the fertilizers, in such cases, we actually don't need to remineralize the water. Good point. Thank you.
And we had a couple more questions come in. Just trying to be fair with people, kind of seeing it in the order I saw it float in. So G Lane had asked, with PFAS being identified in the produced water samples in the soils, is there any way to remove this contaminant from the produced water or the soil to prevent the amount of PFAS in the environment from increasing?
It looks like Pei answered that question. And so what she wrote was that it was detected at 0.4 nanograms per liter, which is below the EPA's recommended limit. So thank you, Pei, for answering that. Yes, it was pretty effective at removing PFAS, which is another reason that we have our polishing step process. The heavy metals question, I don't believe it. I don't remember seeing myself in terms of detecting heavy metals in the plants. I was handling a lot of the forage analysis, and Punhasa was working on a lot of the more high-depth metals analysis. Do you remember anything there?
No, I think we didn't see any heavy ions uptake in the plants as well.
Mm-hmm. Yes, we ran the lab that I was using to analyze the crop outside sending the crop to New Mexico State was called Dairyland Laboratories. So it's a where most farmers will send their hay to check for nutrition analysis, and they'll check for toxins, mold, heavy metals, and different nutrient content. So that's the data that we have in the paper.
Very awesome. I think someone else has their hand up.
Yeah, Tom Merrifield. My question is really around the water balance. Did you have a targeted water balance, or did y'all construct a water balance on this test or these tests? And particularly what I'm curious about is how much infiltration past the root zone that you allowed. That's really my fundamental question.
Mm-hmm. The way that we determined the watering schedule was to minimize the amount of water that was pooling. So in the beginning of the study, we wanted to understand, how much water alfalfa typically needs. So that's what we did some research with farmers to determine that, and then determine based on how much water alfalfa is standard irrigated with, and accounting for 50 to 60% of evaporation overnight, we were able to make a schedule based on how long could we irrigate without having significant cooling on the surface, and how many we had based on the amount required for the plants. I will also better than I think in the efficient, as well as the changing of the growth for the plants. It was less nutrition was the clear though, so that we could observe it. But what we found is that what we would observe was on the side of the box, it wouldn't look like the water was going very low. So, throughout the duration of the project, I think we only sampled all the way down to 3 foot, maybe one time, because, we didn't want to, they're not very large boxes. We thought they were, being 2 by 7, but when you dig that deep down, you do destroy quite a bit of your biomass and the root zones of those different plants. So sampling mostly the top 1 foot throughout, and then save those 3 foot analysis samples till the end.
From an accounting standpoint, did you see any new growth, germination of new plants in the area?
Yes, absolutely we did. In the native plant boxes, actually, where we brought in soil from the field, we did see the mesquite trees did like uptake almost all of the water. So we saw the plants closest to the mesquite trees, not faring well. I think the mesquite trees were just very happy to take up all that water. But we did see some grasses and weeds growing in that soil that we didn't put there. Just, kind of indicating that came from seed that was present in the soil from the field. And the native plant piece, and then on the alfalfa piece, all of that was seeded. So and we would cut before, because alfalfa I believe is a legume, and it makes the seeds from flowers. And so as soon as a plant would start flowering, we would cut it. So there was no, seeds coming from the alfalfa, but from the native plants, there was some in the soil. And we're in our next pilot that I mentioned earlier, that is more rehabilitation focused. There are separate areas some of them are seeded, some of them are not seeded, some of them are, just disturbed. And so we're actually studying that now, continuing to study that on the native restoration side versus the agricultural side.
One final question, kind of comment too, is a couple of decades ago, Herman Bauer was tackling this problem, in the Phoenix area, and one of his recommendations were to use salt tolerant plants. And I think mostly, those found in estuaries or places like that. Is that something that y'all are considering in the future?
Well, alfalfa is salt tolerant, especially there's different variations of it that can handle up to 10,000 parts per million. My only, I guess, reason to say that I don't know that that's necessary is because during the time of this pilot, the Railroad Commission released their pilot standard that didn't allow application of water over 1,000 parts per million for treated produced water, and that has now shifted to CQ, which I believe has a different standard. But I don't know that we would get to a point where we were ever applying water that had a high salinity to the soil.
Thank you much. I appreciate it.
Absolutely.
I thought I saw your hand up earlier.
Yeah, I don't know where it went. Thank you. Hey, Adrianne, question on the technology, please. I think I saw from the freeze crystallization, there's a 70% recovery of the water. That was like slide 6. I don't know if it was higher. 70 to 80. So 2 questions on that is, does that suggest there's a higher limit, upper end limit on how saline the water can be to feed to the salt, to the SWRO? And related to that is, I always had thought water that went through an RO system would come out at less than 500 TDS, and I was surprised to see that there was waters being looked at with higher than what I would have expected. And do you think you can drive this process further with like an OARO or CFRO? So I've got 3 questions in that. I'm sorry to hit you all with that, but that's the gist of what I'm trying to understand.
Yeah, that's okay. Yeah, so I have the 70 to 80%, but when I was presenting this slide earlier, I mentioned that percentage, that recovery percentage is highly dependent on the starting salinity of the water. So where we were testing, when we first started this pilot, we brought water in from all different parts of the field. And so sometimes water came in at about 65,000 TDS, sometimes it came in at about 180,000. So that percent reduction really should be, we get the salinity down to less than 25,000 parts per million. What the volume recovery looks like in order to do that is solely based on the starting salinity and how much that becomes that 220,000 part per million concentrate. This step, so doing freeze desalination the way that we did it in this pilot, slowly on the surface of these tubes, and you have these very large ice crystals, that batch process and the size of the crystal actually decreases your efficiency. So you are more likely to have more salt in your crystal if it's bigger. Some things that we've changed are moving to smaller crystals, different types of slush that can be isolated, and then you can reach that 80% salinity reduction in one pass versus having to do it multiple times, maybe getting 50%, doing it this way, and then having to do it again so that you get down to, say, 20,000 ppm so you can go into an RO. And you are right, too. The RO produced water that was about 200 to 300 parts per million. The water in this study, we actually added some amount of RO concentrate back to get it to that desired range, which was the 500 range, the 1,000, and the 1,500. So in doing that and adding that concentrate back, we were adding some minerals back, and we saw the positive effect that had on the plants. But like I said, we did also observe some concentrations of metals in the soil that we believe was attributed to us adding that RO concentrate back, as opposed to adding specific minerals back, like Punhasa mentioned, in a more of a valorization type.
Great. I'm glad you clarified that. I was kind of curious. And is it conventional SWRO or, we've
Yes.
seen in the last couple of years, OARO, CFRO. Does that play a role possibly in your technology going forward?
I think it can, but we've been using conventional seawater RO. During this pilot, our water coming from freeze desalination was anywhere from 20 to 25,000. Going forward, our water is probably going to be somewhere around 20 to 18,000. I think what we're trying to understand a little bit better in the larger pilot that we're building is where that is from an energy standpoint in terms of how low should you get with freeze desalination before RO becomes a more efficient way to desalinate water. So that's something that we're hoping to analyze in the next phase, where we have less of a batch process and we can actually get a true energy cost and cost per barrel on both.
I'm going to sneak in one last question, please. And the question is, there was some discussion, and I think I saw a question in the chat about organics that were in the soil, and there was some analysis. What I wonder is, was there any analysis of a breakdown of any of that trace amount of TPH or oils by the microbes in the soil, and any consideration given to if microbes are breaking down, whether it's oil or greases, hydrocarbons of that trace level, if they're generating nutrients as well? Has there been any thought given to that?
So we assume that the microbial does help to maintain the organic composition, but we don't know exactly how the microbial contribute to the system. So it's something we are looking forward to study, but at the moment we don't have a clear idea about that. Sorry about that.
I think it's an interesting thing to keep in mind and consider. There have been some oil disasters, as we all know, offshore and hitting the coast, and it's been amazing to many people at how quickly the environment has been able to heal itself. And it's really because the microbes were breaking down, and we're talking about massive situations there. Here, we're talking about trace amounts. And it's just something that maybe in the future you might give some consideration to in some of the academic work you're doing. It's a fantastic program. Please, I think what's been done here is great. I'm just encouraging you to take a look at that as well.
Thank you, Vivian.
Yeah, hi,
sorry.
Joe. This is Pei. Thank you so much for the excellent comments. Actually, that is some study we are also looking at, because in addition to Adrianne and Punhasa presented today, and we are also doing the fit and transport modeling and experimental work using the columns with plants and without plants. And then we are also monitoring, for example, all these organics. It's not all the organics, but selected representative organics in the treated produced water. If this water pass through the columns and with microbes, what will be their biodegradation or their retention in the soil. So hopefully we can answer the question soon. And as I also posted in the chat, we have also the non-target analysis ongoing right now, and we collected the samples from this greenhouse study on the soil samples and also plant samples. Once we get the non-target analysis results, I think we can also in a better position to answer all these questions. So with that being said, so Joe, we really appreciate the comments and also from the backlayer. So that is ongoing actually.
No, I appreciate that. It's been fascinating. We did some work in my past time with Oxy, and it's fascinating to see microbial communities and how they change. So yeah, I think it's important to look at beginning and what happens. It's not just one microbe, it's not two microbes, and it's an entire family, and they all feed off each other and live together. And I think it'd be fascinating to look at that and what are the degradation products, and are they truly becoming nutrients? But thank you for that, Pei. Appreciate it.
Thank you, Joe.
I'll answer one of the chats. It says, have you done a cost benefit analysis of using the permeate plus the fertilizer versus the permeate plus the RO concentrate? In terms of cost analysis, no, we haven't done that. But in thinking about it, the permeate plus the RO concentrate, I would think would be more expensive in that scenario, since the concentrate is going to need conditioning and treatment prior to being able to add it. I think that, that shouldn't deter us from looking to that RO concentrate for different minerals that we could extract and use for fertilizer later on. But if you were going to just go and start a project, I think right now it would still be cheaper to use fertilizer.
And I think it's also important to include the deep well injection cost as well for the produced water, because if we utilize this water for other application, we will be saving some from the cost that we have to associate with removing these produced water from the system.
Yeah, quick question off of that, Punhasa. What was the limiting constituent in the RO process? Was it just the salinity overall, or were there like silica or hardness that prevented a higher recovery?
Can you ask that again, Zach? I think I missed the beginning of it.
Yeah, sorry about that. I was just asking about, what was the limiting constituent in the SWRO? Just thinking about, being able to increase water recovery, we're thinking about divalents, like, oh, if you could precipitate those out or somehow selectively use those as a fertilizer instead of using the entire concentrate stream, but what would, is it possible to increase the recovery on that SWRO unit?
I think it absolutely would be if we were able to remove more of those divalents. The green sand filter did help a little bit. And we had that because we knew that there was potential for iron and aluminum to be present. In the membrane analysis that we did on these membranes post piloting, the primary scales that we saw were silica and aluminum. So that was kind of surprising to us. We thought we were going to see more of, calcium, magnesium, barium type things, but we actually didn't. It was aluminum and silica scales that were observed.
Very interesting. Did you use anti-scalant?
No, we didn't. We didn't
wow.
at this size. But on the larger project, the 10,000 barrel a day one, that project is going to have anti-scalant specific to those things that we saw in the last study. We actually do have another paper, a separate paper that was written about the specifics of the RO process here. I can share it with you and you can distribute it.
That would be amazing. Thank you. All right, what other questions do we have? Going once? Going twice? Okay. There was a lot of great questions, and so thank you, Joe. Thanks for everyone for participating here. There was a last question here, but it was just about the recording. Is it going to be available on our YouTube site? And yes, of course they will. They will always be posted there. Give us about a week to turn that around, and we're aiming for before the holidays to get this out. But if there are no other questions, no other comments, I'd like to thank our presenters here. And yeah, thanks for sharing your time and your expertise and for joining us today.